So without further ado, I would like to hand over to you, Professor Jidong Gu, Professor at Guangdong. I'm sorry if I'm going to murder this pronunciation here. Guangdong Technion Israel Institute of Technology.
Yes.
And you're going to talk to us about microorganisms and world cultural heritage. It's a pleasure to have you here. Please take it away. The floor is yours.
Thank you very much.
No problem.
Yeah, my name is Jidong Gu. I'm working at Guangdong Technion, Israel Institute of Technology in mainland China. And today I'm going to present a talk on microorganism in line of International Microorganism Day, and also the relation to World Cultural Heritage. And this work has been a long time accumulation of research evidence with Japanese colleague Yoko Katayama, and that's why I put her picture over here, because we have collaborated more than 15 years on this project. So to start with, I'm going to give you an outline what I'm going to talk about in this 15-20 minutes. I'm going to introduce briefly about the subject of cultural heritage and biodeterioration. And I'm going to focus more on stone material as the material of cultural heritage here. And I then going to look at the microbial processes which are affecting the integrity of the stone material, which is cultural heritage. And then several specific processes which are damaging the sandstone. So basically, that's a combination of science and also tune down of some of the information to a better understanding, I hope. So to start with, when we talk about cultural heritage, especially in Southeast Asia, Angkor Wat. Angkor Wat is probably one of the most famous cultural heritage. And it's so famous. And over here, you can see it's Angkor Wat. And in front of this small pond with reflection, magnificent. And it's also being shown on the national flag of Cambodia. This cultural heritage, if you get closer to it, it will be able to show to you that there is a constant maintenance, repairing of the cultural heritage because it's old, about 1,000 years history, and it has some maintenance work all the time. And this particular cultural heritage has been listed as World Cultural Heritage. It has about 18 years of history. It's not that long. 2 years ago, we had a celebration of 25th years of cultural heritage for this Cambodia site, which was held in Angkor Wat in Cambodia. So if we look into the importance, historical importance of Angkor Wat, it's really the culture, humanity, the art. And this is one part of the Angkor Wat. If you go there, look at the bas relief carving. The carving is on sandstone, and the carving on this stretch is 400 meters. It is an enclosure of 4 sides. So basically, if you walk around 400 meters in the east, 400 meters in the west, and if you go around it, it's 1,600 meters of bas relief. And on this carving is recorded history of Cambodia, which is a Khmer in the past. So it has a great importance of humanity, of artistic presentation, and also the humanity of that particular culture. But now I'm shown to you at the early part was about Angkor Wat. Angkor Wat has been the international image for Cambodia, but actually closer to the Angkor Wat, there is Angkor Thom. What does this Thom mean is this is Angkor city, because Angkor Wat is a temple, but Angkor Thom is a city. The city was a real live city. The king was living in it. There were also people living in it. But Angkor Thom has not get so much importance and attraction as Angkor Wat. So I want to show you some information about Angkor Thom. And this is going into Angkor Thom, the city in the south side, south gate. If you look at on the left-hand side and also similarly on the right-hand side, which you can't see, they are statue of guard, and they are the warrior guard. But below it is canal, water. So the water was and actually is still surrounded the city, but on top of the river, the canal, there is a gate. So we can walk and we can go into it. And within the city, it is where the life and the king would be. So if we go into one of the temple in Angkor Thom, this is called Bayon Temple. And for the Bayon Temple, you can see a little bit, but actually this is the central tower. And if you look at the appearance of the sandstone architect, with all different kind of microbe, that's the color about. But if you look at this particular stretch of section, it's a gallery. The wall are all being carved over here. And you can also see over here, this particular section of the stone column, the top part was missing. So immediately, this is in the 1960s, when the French discover Angkor Wat again, they use concrete to make an emergency protection and conservation of this part of the gallery. But if you look at a little bit to the left here, you can see here, this section of the tower, the top part was missing. Missing in this part was missing. So this particular gallery, this section is completely exposed. So the carving are exposed. So similarly over here, this part of the tower is also missing. So what I'm trying to show here is the temple has a long history and is also in a damaging state, being destructed. And when we look at the magnificent part of the Bayon Temple, it is on top of the tower. On every tower, there are 4 large faces of the each to the cardinal direction, and each of them are unique. There is no duplicate. Every one of them are unique. This gives the major attraction of the Bayon Temple to tourists. So what we do, when we started working on protection of Angkor Wat and Angkor Thom and also other temple, we can go to the temple and do sampling. This is something I'm going to talk about in terms of technically how we did it. After we get our sample, then we bring back the sample to the lab to analyze what are the microorganisms associated with this brick red color, this dark color, and here is a brownish color. By doing that analysis, we would be able to know what are the microbes are living here on the surface to cause this discoloration of the bas relief carving on the temple. Something I want to briefly mention here is I'm not going to be able to go to great detail is water in the protection of the temple. So in another way, if we can limit the amount of natural water, rainwater to the temple, then Angkor Wat would be able to be protected for a much, much longer time. The second factor in this whole process, this is something I'm going to be into more detail, is the biochemical reaction of the microbe, which are destroying the temple. They contribute to the damage of the temple we are seeing today. And this is the second part. And the third part is a summary, which has just been come out this week, actually. This is a publication we have wrote, and this is summary of what is happening on Angkor Wat, on the sandstone. If you are interested in, you can get the article, and then you can read some of the information in more detail. So the first step is to collecting the sample, which would not destroy or damaging the sandstone of the temple. So the technique of sampling. This is a technique which has been invented, and which has a adhesive material inside, but the whole sampling device is sterile without any microbe. So what we can do is we can open this device, let the adhesive part to adhere on the surface of the sandstone, so any sandstone. And then if you look at the illustration on the right, which would be a cross-section of this sampling device, push onto the biofilm, the microorganism, then we can peel it off. This is the first sample. Afterward, we can take a fresh new device, push it onto the biofilm again, we take the second sample. So in theory, we can take the third one, we can also take the fourth one. And basically, on this particular site, you can take as many as you want for the sample. And after taking the sample, each of them going to be in this device, we take back into the laboratory, we can extract the DNA of the microorganism, amplify them by PCR technique. Afterward, we can manipulate basically to reveal this particular sample. This is over the depth of the sample. So initially, this is the sample we take, and then we analyze. And here is look at one particular site over here. We take 36 sample, and over time, we will be able to analyze the DNA in the sample. We see dynamic change of different microbe over the depth, because the first one is the outer surface, and when we go into higher and higher number, that means into the deeper of the biofilm, you can see that cyanobacteria was high in the outer surface. When we get deeper, their number are get lower. But the facultative microorganisms, which do not like oxygen, they become higher and higher in the lower layer. So we are able to get the spatial information of the microorganism distribution in this particular site and on the sandstone. This is one of the techniques. So similarly, we can also get the whole community composition of the different microorganisms. This is at one particular site of the biofilm 42. And by doing in the community, but there is also some problem. Which one are actually damaging the sandstone? This next generation sequencing does not tell us. We need other technique. So let's look at one of the example is the sulfur. This is at Angkor Wat. You can see on the back part is the bas relief, and here is one of the column in the gallery, and the lower part has been damaged. So what we did was, this is the Yoko Katayama. We took a sample on the top without any damage. We took a sample at the intersection between damage and intact. And then we take sample at the serious damaged part. So there is A, B, and C. So for the 3 sample we took, we incubate them in the culture media for sulfur oxidizing microorganism. What we see is within 5 days, C, which is the very, very lower part, they would have a pH drop of 1.2. This means production of high acidity, a lot of acid being produced. But for the B and A, even after incubated for more than 2 months, the pH drop was minimal, very little, particularly for the top part, there is no pH drop. So this indicating sulfur transformation are involved in the damaging of the sandstone, producing acidity. So similarly, if we cultivating the microorganism, this is the bacteria we isolated. You can see over here, if we put sulfur in, the microorganisms grow very well. If we do not put sulfur in, they do not grow that well. But when they grow very well, they cause a pH drop. This is the pH drop. And here is the control. The pH doesn't really change that much. And similarly, if we monitor another group of microorganisms, which are fungi, Angkor Wat, Angkor Thom, and also non-chrome, which are 3 different sites, you can see that the fungal, which can oxidize sulfur, their population are between 100 to 200 per gram of material. But this is a culturable fungi, which are much less than the total number of fungi, but we found them on the temple. And here is also an illustration in the laboratory condition. We can see that the microorganism can utilize the sulfur in the agar plate, causing a clearing zone. If we analyze what is over here, the C section, we found they are thiosulfate and sulfate. So those are the products from transformation of the sulfur put onto the agar plate. This is a complicated process, but I'm not going because of the time. I'm only going to touch onto another process. What we have found recently is nitrogen transformation is also evident. What we found was when we monitoring what is in the moisture or the water of the sandstone, we found there are higher concentration of nitrate, but not ammonia or nitrite. This leads us to monitoring what is happening here. So this is related to the nitrogen cycle. I'm going to jump quicker a little bit to save my time. So what we did was we take sample at the ground level, in the gallery level, and also the higher level of the Bayon Temple. We analyze what are the microorganisms in it. So on the ground level, gallery level, and the central tower, we see much higher concentration of microorganism called ammonia oxidizing archaea, much higher than bacteria. What does that mean? Both of them are contribute to nitrate and also acidity, but archaea concentration are much higher. That means archaea are the driver of this reaction. We know what are the microorganism name, bacteria name, and we repeat this particular experiment in the following year at 3 different temples. We found similar result. That means archaea are much higher than bacteria. And we also carry out a simulation experiment, and we carry 36 liters of deionized water onto the top part of this gallery. Sorry. And then we simulating rain water. So let the water to drip down and collecting the dripping down water and then analyze. We found there are ammonia, nitrate, there are also a lot of sulfate in it. So those are the iron which has been leached out from the sandstone to confirm what we have observed previously. And the last piece I want to show to you is we use another technique, which is called isotope technique. This allows us to differentiate what are the 2 processes contribute to the removal of nitrate. By doing it this way, I'm not going into the technical detail, but I want to show to you is there is a very active ammonia oxidation to produce the nitrate removal by denitrification and the animal process was very weak because I use a small arrow here. And because of that, the nitrates are being accumulated because they are not being removed very quickly by the 2 processes. This is what we found on Angkor Wat, which result in accumulation of nitrate on the sandstone. So basically, if I conclude, I would say that colonization of microorganisms on the surface of a sandstone monument is a common phenomenon. We cannot prevent this to occur as long as the sandstone monument is in the open environment. When water is involved, particularly in tropical environment, microorganisms going to grow very actively, and the damage to the sandstone by microorganisms would be higher. The damaging process in this particular case we presented involve sulfur oxidation, ammonia oxidation, and in the ammonia oxidation process, it is the ammonia oxidizing archaea play a much more important role than ammonia oxidation bacteria. So to understand the damaging process causing biodeterioration of the sandstone, both biology are important, which is providing the basis for our understanding of the process involved, and also providing the information for management of the Angkor Wat in the long run. Because my time is running up, I would conclude that this project has been supported by Japanese trust fund for a long time, more than 15 years, and recently has also been supported by the Research Grant Council of Hong Kong government. So with that, I would just stop here, and if there are any questions, I would try to entertain any question, and that's the end of my talk. Thank you very much.
Thank you so much, Professor Ji Donggu. Unfortunately, we don't have time for questions just now, but if there's any, is there any way for someone to reach you to ask questions later on?
Yeah, sure. Because on my first slide, I show my email, and anyone, if they have questions, they can contact me through the email. That would be one way. To my email or through any means, because for the Zoom, I would also afterward monitoring if there are any questions sent to this particularly event.